Hello, everyone. This is chapter 2 in your textbook, and we're going to begin speaking about molecules and cells today. So we're going to go into the understanding of chemistry, really, and how it builds biology and then physiology. In truth, it all begins with physics, energy. Your body is designed as a giant transformer or energy transmuter. That all begins with the physics of things like, I said energy, but frequency, vibrations, all matter. Remember these, if you've ever heard these phrases, they hold very true. Matter has property of both particle and wave, means yes, it sits still. Okay, that's how we have like tangible reality around us, but it also absorbs and emits frequencies. So actually everything's like always in motion. Also, the equation E equals mc squared, that says energy equals matter times the speed of light. So energy is truly not separate from the matter or material that we are used to. So those foundations of wavelengths and frequencies, different kinds of energy fields and types of energy and spectrums of energy will be generated as well as absorbed, reflected, or refracted by your body, by the human body. What happens when any of these energies get inside, and what I mean by any of this is you can either eat food, okay, then we have chemical energy that's going to be transformed into mechanical energy, meaning you're going to take the energy you get from food and then you're going to go ride a bike or something. This also happens when we talk about electromagnetic frequencies. You get sunlight that hits your skin that produces vitamin D in your body. That is electromagnetic energy that's being transformed or converted into chemical energy. And then that chemical energy is going to go do some mechanical things. So the human body is pretty awesome with the transformation of physics into chemistry, into biology and physiology. What I think we have to understand from that platform then is that if all of that is true, it's all really linked into what our bodies will do and are able to do. Also, it goes into our behaviors and our actions and stuff like that. So at the very, very foundation of your body is physics, things you can't even see, things that your body is interpreting, but you may not be consciously aware of or how to, or that it's even happening, but your body does. Your body's picking stuff up all the time. So we're going to go into just the discussion about atoms and the forces. So when we talk about the atom, if you remember this is just some foundational chemistry. It is a fundamental building block of everything. It is classified. What we have done in humanity is taken things that we can observe and we classify them, right? That's kind of what we do in science. So the pieces and parts of an atom are the protons, which have a positive charge, neutrons, which have no charge, and then electrons. So if you remember this somewhere from a different biology class or chemistry class, the protons and the neutrons are really what give an atom its atomic mass. And they sit in the nucleus, in the center of an atom. And around that will spin electrons, which then creates this energetic field around the atom. The electrons will circle around that nucleus in specific orbital patterns. And really, it's the electrons that deliver an atom a lot of potential, what it's going to be able to do with other atoms. Okay, it's reactivity. Does it want to dance with another atom or does it not want to dance with another atom? Again, what I mentioned about the nucleus of an atom is that those protons and neutrons are going to be primarily the bulk mass of the atom. It's good to note there's a difference between mass and weight. Mass is how much matter is in there. Weight is mass with gravity applied to it. So for example, if you go to the moon, you're going to have the same amount of mass, okay, density to you, matter to you that comprises you, but there's no gravity on the moon, so you don't weigh anything, if that makes sense to you. When we talk about chemical bonds, okay, this is where the electrons circulating within certain orbitals outside that nucleus come into play. They, again, will dictate an atom's potential reactivity with another atom. So an atom may want to bond or link up with something else to form what we call a molecule. So in this example, we're talking about this is a water molecule. We have oxygen in the center and two hydrogens that look like they kind of swing towards one end of the molecule. And these are different models or maps or formulas. We're going to look at this so we can see an entire molecule and then investigate the atoms within it. Within water, what kind of happens is water has more of a negative charge just by itself, okay, due to the electrons in one of its shells. And then the hydrogen atoms are gonna kind of gravitate toward the other pole of the molecule and hold a more positive charge. I'm mentioning this in regards to water because this is part of the reason water has some of its properties. It's how these charges and polarities arrange themselves within a molecule, deliver it its properties, and how it will react with other water molecules and other things. Now, on this picture, you can see, let's take that oxygen atom, for example. The big red ball, that's the center, that's the nucleus. Remember, there are protons and neutrons in there, and generally they're going to balance one another out, not always. Okay. And then you'll have what's called valence shells or shells of electrons. Okay. The first shell where we find electrons orbiting outside of that nucleus, well, you always hold two electrons will spin out there. Or to fill full, it will want 2 electrons in that first shell. All of the shells outside of that are going to want to hold 8. That will make those shells happy and balanced, and they're going to feel good, okay? If, and this happens often, if there's an atom that doesn't have its outermost shells or valence shells of electrons full or satisfied, it will want to find a dance partner. That's kind of how it works. Now, when we're talking about formation of molecules, we have to be aware of different types of bonds. Different types of bonds between atoms or even between molecules to form something bigger called a compound. They have certain properties. So the first type of bond we're going to talk about between atoms or molecules is a covalent bond. This is when 2 atoms collectively lower their combined energy. This is a very strong bond, and it's not easily pulled apart or dissociated. An example of this would be the atoms of amino acids, sugars, and fats. In a covalent bond, okay, you can have 2 different types of bonds within this category. One called a polar covalent bond, or a non-polar covalent bond. Now, if you just look at the terminology of polar versus non-polar, if something is polar, it has opposing forces at opposing poles, like the North Pole and the South Pole, negative, positive. masculine, feminine, that kind of thinking. Okay, that's a polarity. Okay, for when we're talking chemistry, we're talking positive charge and negative charge. Okay, positive charges are the electrical force, whereas a negative charge is a more magnetic force. So when we talk polar covalent bonds and nonpolar covalent bonds, in a polar covalent bond, we have a polar situation. We kind of have opposing charges at different poles of our molecule. And what will happen in that situation, if it's polar covalent, there's going to be an unequal sharing of an electron. In a covalent bond situation, there is a kind of a sharing of the electrons. And one of the molecules, or I'm sorry, one of the atoms within the molecule will pull on a spinning electron a little bit more. So it's like, pretend you have two atoms that in their valence shell, they're not balanced. They're not happy. So they're going to want to dance. They're going to want to go on a date with another atom, and they're going to want to join their forces together. So we'll pretend the electrons spinning around them are children. Okay. So in a polar covalent bond situation, there's kind of an unequal sharing of that valence electron. Okay. It's like unequal custody of the electron. One of the atom parents gets to see the child more frequently. In a nonpolar situation, this again is a shared type of bond, but it's nonpolar. So the difference in the charges at the ends of the molecule it's not as big. Therefore, any electron shared between the partners is going to be a more balanced sharing. Ionic bonds are pretty strong. These are due to an electrical attraction. We have cations, positive charge, which is more of an electrical type energy. And then we have anions. Anions are your negative, more magnetic part of the force. So ionic bonds, again, pretty strong. An example would be salt, sodium and chloride. They have what we call an ionic bond. And in this, it's more of a transfer of an electron from one of the partners to the other, and then they're kind of glued together because of the transfer. In a hydrogen bond, this is a style of bonding that's not as strong as covalent or ionic bonds. It is a type of what we call a van der Waal force. It's easily broken and it can be reversed. An example would be proteins interacting with one another, but also water has hydrogen bonds. So water molecules that want to come together, okay. Again, this is part of the reason water has some of its properties, but it's also the reason that water strider can hang out on top of the water with its legs not penetrating these types of forces. In our DNA double helix, we also have evidence of different types of bonds. So in this picture, this is a very nice picture that demonstrates this well. When we have these two pieces of this network that get hooked together in order to form this double helix, the bond between the nitrogen bases, the rungs of the ladder, the double helix, are hydrogen bonds. The rest of the bonds between atoms and molecules are covalent. So the hydrogen bonds are a little bit weaker than covalent bonds. We just learned that covalent bonds are strong. Hydrogen bonds are not so strong. So when the DNA replication process take place, this is a benefit because what will be broken apart are the hydrogen bonds in the center of the rungs of the DNA ladder. When we discuss isotopes, an isotope will have an equal number of protons, but a different number of neutrons. So for example, you can have a carbon atom, which has very similar properties. Electrons are the same number, but the neutrons within the nucleus are different. So picture the atom, right, in that nucleus in the red ball, we have protons, positive charge, which we want to have equal the amount of electrons that are spinning around it to balance the charge. The neutron, usually in its simplest elemental form, will equal the number of protons. However, in an isotope, this is not the case. So we have carbon 12, carbon 13, and carbon 14. And we have found this useful information for like carbon dating. Okay, if they become, if they're radioactive, they will be unstable. And we'll be able to use it to identify age determinants of like fossils and stuff like that. So when we talk about different types of molecules, remember we went from the atom, we linked them together in a certain kind of bonding way. We've made a molecule. Now there are different kinds of molecules. As we break apart this class, they can either be organic molecules or inorganic molecules. Now, I will say right here that your organic molecules are going to contain carbon chains or carbon rings. They're going to be bigger. They're going to be more of your macromolecule group. Your macromolecules are going to be your proteins, your nucleic acids, your lipids. Did I say carbohydrates? And your carbs? Carbs, proteins, nucleic acids, and lipids. Your inorganic molecules, these are going to be more of your minerals and your ionic compounds. When we break apart or begin to get into the classification of the different types of molecules, we have sugar and starch molecules in the carbohydrate category. Within carbs, we have simple carbs or complex. Okay, well, we are talking, and you've heard this likely all over the place, when we are beginning to discuss carbohydrates and their quality, it has to do with how big the molecules are and how easily they're broken apart and how easily their energy can be harvested. One of the roles of carbohydrates, I think we know this, is because they're easy to access their energy. And we utilize simple carbohydrate in the form of glucose within every cell in order to break apart bonds, harvest energy, and utilize it within cellular mechanisms to produce, like, more cell walls or build more fats or build more proteins. So we access the energy within the chemical bond in order to do work within our body. In a simple carbohydrate situation, examples of that would be glucose and sucrose. Complex carbs, these are polymers or glycogen. Now, a polymer is a big long chain in comparison to your simple carbohydrates. When we talk about carbohydrates, also, it's just some clues of language. When you see the suffix OSE, you are talking about a sugar. You are talking about a type of carbohydrate. Within this category also, we have to recognize that we can have monosaccharides, 1 sugar, a disaccharide, 2 sugar. Saccharide is also an indicator that we're dealing with a sugar, i.e. a carbohydrate. You can have a polysaccharide. polysaccharide as well. That's a polysaccharide, poly meaning multiple or many. Okay, you're going to have many sugars in a linked chain. And again, this is an organic molecule. A carbohydrate therefore has long linkages, either in chain form or ring form of carbon molecules, and then it'll have different things around it. So when we talk the simplicity of things or simple carbs, that's what we might be looking at. An example of a polysaccharide would be starch or glycogen. When we talk complex carbohydrates, these will be your bigger saccharides or your sugars. I think I said that right. Again, used as a fuel source, we're going to access and harvest the energy within the chemical bonds. When we talk about our second grouping of organic molecules or macromolecules, these are the proteins. Proteins are long chains of amino acids. So let me back the train up 1 second and clarify this language. So on this carbohydrate slide, under the word complex, carb, you see the word polymer, and then we do see the word glycogen, right? I mentioned glycogen being a polysaccharide, big long chain. So, and I hope I said that right previously, but let's talk about the polymer. A polymer is a big long chain of something, okay? We have monomers, which are the simplest form of our macromolecules or our organic molecules. So the simplest form of a carbohydrate that we can get, or that we're after, or that we would want to break it down into, okay, would be its monomer, which is glucose. That's the monomer of all the other carbohydrates, saccharides, or sugars of any kind. We want to pull it down into that monomer, which is glucose. So back to proteins. Proteins is the polymer, meaning that we take monomers, simplest form of a protein, which is amino acid, and we link them together. Okay, so when we talk proteins, also you might say language like peptides. That references this category of organic molecule. If you have a dipeptide, you have 2 amino acids linked together, and they're called that because of the kind of linkage that they do. You can have a polypeptide, which is going to go probably somewhere from 15 amino acids to 50 linked together. Once you get over 50 amino acids linked together, now we classify them as protein. Proteins have tons of functions. You can break down proteins for energy. You have a different metabolite from that, okay, due to how, the amine groups and the amino acids. But proteins, they act as carrier molecules for some things. And they also help catalyze biological reactions, meaning when we catalyze something, we are saying that we are reducing the amount of energy required to make this chemical reaction happen. Therefore, it makes it happen faster. So here's an example of what an amino acid would look like structurally. So structural formula. On the end, you'll notice what we call the amino group. That's the N with the 2 hydrogens off of it. In the center, that's our central carbon, and you'll also notice the R in the box. That's our R group in amino acids, and it's one of the identifiers. That R group will be the identifying factor between different amino acids. That's what's going to give one its quality and or property over a different one. Then we see the acid group on the end. Okay, here's where we talked about peptides and peptide bonds. I already mentioned this. This is really important. I think this part's one of the most important things for us to understand as we move forward in our understanding of the human body. It's the structure and the shape of proteins. Protein shape will determine its function, what it's able to do. So we classify proteins according to 4 structures: primary structure, secondary structure, tertiary structure, and quaternary structure. The primary structure of a protein is like a bunch of amino acids just in a line, like a choo-choo train. They're straight. As this begins to get bigger, though, as this row of amino acid linkages just keeps adding on to itself, it's going to start to twist a little and go into what's called a secondary form or shape of a protein. Check the secondary structure. So it will begin to have linkages, bonds, and reactions within itself. So it starts to twist. So the shape or form that should be associated in your mind when we're talking secondary structure of proteins should be what's called an alpha helix, one ribbon, okay, or what's called a beta pleated sheet. And there are examples here of the different shapes. Now, as these guys begin to build and grow and get bigger, more amino acids linked, it's going to take on what's called a tertiary structure. Tertiary structures are what I call balls of yarn. This thing's really wound up on itself. Now, this is super relevant. Tertiary structure proteins are our globular proteins. These are going to be used for our antibody makeup and a ton of other things, but that's just a great example. Now, when we have these tertiary structures, these balls of yarn, they start linking together. When you have 2 or more tertiary structure or globular proteins start to link together, you're going to have what's called a quaternary structure. So an example of a quaternary structure protein is hemoglobin. Super relevant. If it doesn't maintain its shape, it's going to lose its function. So if you think about it, put that in context of what I just told you. If you have an antibody that starts to disassemble itself, okay, you're going to have an impaired immune function. Now, what would disassemble that protein? Extreme temperatures or out of control temperatures. That doesn't always mean a fever. That's metabolic heat production or some type of other energy needing conversion within your body that deranges the protein, some kind of other stimulus. The other thing that will derange that protein is an aberrant pH. If your body is not great at maintaining pH, or you have a highly acidic environment or highly basic, it's just easier to go acidic than it is to go basic, you're going to start ripping proteins apart and they will not function as well. Same environment is going to hold true for our ability to produce and maintain molecules of hemoglobin. If we don't have enough hemoglobin, we can't carry oxygen. If we can't carry oxygen to tissues, it dies. So glycogen, I mentioned that as complex carbohydrate, polysaccharide. Okay, it's a polymer also, big thing. Glycogen is one of our energy storage molecules. So if you're doing like a fast, in order to maintain blood sugar levels for mostly your brain and other things, you need that glucose level to be within a particular range so that metabolic processes can take place and organ systems can function the way they're intended to. So within that, you will access energy from the molecule glycogen. You'll break that bugger apart. So if you have trouble storing glycogen, or I'm going to go a step further here and accessing glycogen due to other things being in disrepair, your blood glucose levels will be dysregulated. And that can come from a variety of origins. That can come from liver congestion and or dysfunction. That can come from pancreatic disruption. Some of the functions of proteins in our body, we just meant some more functions of proteins in our body are, carriers for things, receptors for things. They do produce or are collagen. Collagen is a protein. They make enzymes like salivary amylase. If you don't have enzymes, you're not catalyzing reactions. You're not assisting in the breakdown of things. If you can't do that, certain chemical reactions aren't taking place, or they're going to deplete energy stores. They also function as myoglobin and hemoglobin, as I just mentioned previously. Also that immune system portion. I also want to mention this. The function of your DNA, what it does, it holds the code to make proteins. That's how important all of that is. Without the proper code, without the proper reading of the code, without the proper production system and resources to build proteins, you don't build proteins. You don't repair things. You don't repair cell membranes. You don't carry hormones or neurotransmitters well. You don't break down your food. Therefore, you don't get your nutrients. So this cellular portion of what I'm teaching you here is fundamental to understanding really the origin of disease processes. It is at this microscale level. I feel if you get a grip on it and understand this, you can figure out almost anything, because this is where it's going to begin to go wrong. And then you can figure out how to course correct a little bit differently. So when we talk about lipids rich in carbon and hydrogen, they do contain oxygen. They do not dissolve well in water or blood. And lipids, yes, that's fats. You need them in your diet. So, and just because you eat a lot of fat doesn't mean you will gain a lot of adipose tissue or fat in the body. That is not what that equates to. In fact, learning a functional way to have, lipids as part of your diet can help you actually clean up a lot of processes in your body if it's done with some conscious awareness. Fatty acids, hydrocarbon chains with carboxylic acid group at one end. We hear a lot about this. Saturated versus unsaturated fats. Example, stearic acid and oleic acid. Okay, I'm going to stay away from stearic and oleic, but I'll plug in my own little thoughts here. We hear a lot about saturated fat gets a bad rap, primarily from animal sources. So a saturated fat at room temperature will be a solid, but it has a higher heat tolerance too. Unsaturated fats, these tend to be from animal sources. Now, when we talk about what this even means, it has to do with the bonds between the carbons. So a saturated fat has single bonds between the carbons, and the entire molecule is lined with hydrogen atoms, which those hydrogen atoms become pretty volatile and can be ripped off easily. When we have an unsaturated fat or fatty acid, the carbons can have double bonds between them and less hydrogens on the outside of the molecule, therefore a little bit more stable of a molecule. Here's what I want to say though. Not all saturated fats get a bad rap, but they're not all created equally. Even if they're labeled as such, you have to understand where it came from, how it was produced. For example, coconut oil might be labeled as saturated fat. It does come from a vegetable source, right? We would perceive that as unhealthy. Not so. Plus, the labeling of it, even though it's saturated, it's like 51% saturated and 49% unsaturated. So yes, it tips the scale, but they're finding more and more out about the. I think it's really an antiquated way of thinking about fats, where the saturated fats are just blanket statement across the board bad for health. That's not accurate. It really goes deeper into origin, where to come from, what did it go through. Also, we can plug in, trans fats. We hear about that. What a trans fat is, it does have to do with the molecular shape and its stability within the body. They are not good because they do tend to kind of rip things apart and set free some free radicals within the body. They break things down that should not be. Also, if you get a blood draw and someone gets a lipid panel done and they have triglycerides in the blood, just good to point out here that high triglycerides in the blood are not indicative of a high fat diet. They're more indicative of a high carbohydrate diet. Too many carbs, and your body's trying to store them, put them into packets. It converts them into energy storage packets. I think we understand that, fats have a higher yes, they're higher in calories. And they store more energy per calorie. So it's like, 9, I guess the measurement is kilocalories, right? Per molecule of fat, in comparison or contrast to that of carbohydrates. When we talk fatty acids, we do have to look at how that molecule is built. A glycerol molecule can have 1, 2, or 3 fatty acids attached. You get mono, glycerides, diglycerides, triglycerides, like I mentioned, and they are for energy storage. They're energy storage packets by the body. This is super relevant. Within your lipid category, you're going to have something called a phospholipid. These little buggers are magic, and they are part of your every cell membrane. So again, here we're putting the plug in for the presence requirement of fats in your body. It is necessary. This is part of the reason why. Okay, within a phospholipid, you have what's called the little square at the top here. Is it the head of the molecule? That's the phosphate part. It's hydrophilic. What that means is it loves water. If something loves water, it's going to be at the polar end of the compound. That is highly relevant in understanding how a cell works, how polarity works, how electricity in the body works. Anything that will dance with water, dissolve in water, has to be polar. If it's polar, that means it's charged or has a charged portion to it that will mix well. This goes back to the principle of oil and water not mixing. Oil, lipid, will not mix with water because there's an absence of polarity within the oil. This is a great example. The little tails here on this phospholipid are what we call hydrophobic. They're afraid of water. They are non-polar, whereas water is polar. Water will break down other polar substances. Within the lipid category are our steroids. This is hugely relevant as well as we talk about health, okay? Within the steroid category, we have cholesterol, estrogen, testosterone. Now, cholesterol, it is the foundation of every hormone within you. So, I think we need to take great care as we're measuring cholesterol levels in a lipid panel via a serum draw and then approaching that appropriately. If we understand now that phospholipids and part of every cell membrane, not as a fat. If we understand that cholesterol is the foundation for every hormone in the body. And part of every cell membrane, by the way, cholesterol molecules help anchor the cell membrane, give it a little bit of rigidity. So as we measure cholesterol circulating in our serum, okay, in our plasma, then we have to understand that perhaps something is wrong, either in our hormones. Now that doesn't always mean our sex hormones, okay? That's a precursor of other things as well. But maybe something's wrong in the cell membrane of our cell because we have a systemic inflammatory process going on due to an immune response. I think that when we get those lipid panels done, if we find aberrations that we're not comfortable with and they're outside of ideal range, I think we need to go diving in some different directions. So the body stores chemical energy. I mentioned that your body being that giant energy transmuter, it holds energy in chemical bonds. So I have this atom that wants to dance with this atom. When they bond together, they are holding energy within that union. When they break apart, okay, that energy goes somewhere else, hopefully used for positive things. Nucleic acids. Nucleic acids are the building blocks of DNA. Okay, nucleic acids, the monomer of a nucleic acid is a nucleotide. The subunits of this molecule, all of them will have that nitrogenous base. That nitrogenous base is what makes the rung of the ladder in a DNA double helix. It will have a sugar, either ribose or deoxyribose. And then it's going to have a phosphate group on it. So here's our DNA molecule. Again, you see the letters in the center here, okay? Those are comprising the rung of the DNA ladder. Okay, those are our base pairs, our nitrogenous bases. Now you have two groupings, purines and pyrimidines. DNA molecules have certain nitrogenous bases within their group, and RNA molecules will have different nitrogenous bases within their group. So when we talk DNA then, or these nucleic acids that have been, which are the polymers of nucleotides, we are aware that they hold our chromosomal and or genetic information. So chromosomes deliver us our genetic code, our genetic map, what's going to happen. In humans, in your somatic cells, which is every cell in you except for your egg and sperm, each one should have 46 chromosomes, 23 from each parent. Red blood cells don't have them. They don't multiply. They don't have a nucleus. So the human genome, that is your genetic map. It is similar in every human, meaning they have determined the locus of particular traits or what each gene will manifest and or translate into as far as characteristic. I mentioned that the nitrogenous bases were slightly different within RNA and DNA. And that is because DNA holds the map and RNA does the translation, or well, let's say transcription, translation, and then protein production from the map. And it's quite an intricate process that begins within the nucleus of a cell. It's fascinating to me that it happens, and also fascinating that we know it happens. This and when you think about this, you guys, you need to understand, number one, if I have not already said this, we do not know everything about the human body. We know what we know today, and good science will expand upon that. All of this information that you're getting is, some of it's really not all that old. The technology is definitely not that old, only within the last few centuries. Well, you put that into comparison of how long we've known, humanity to exist, that's quite a big deal. So what we see utilized either in applied scientific procedures as far as diagnostics, or the technology of, treating the human, these are new. Okay, a lot of our current medical model is based on a lot of very new information in the grand scheme. So we are still part of, the learning process as we learn more about ourselves, how we're built, how things work, and it will change. It should change. I hope it changes. We'll learn more. And hopefully the science, our methods of studying and evaluating this stuff can keep up with the evolution of the human physiology, which should change and probably has already. Hopefully we're keeping up with that. So when we talk about different types of RNA, there's messenger RNA, transfer RNA, ribosomal RNA, and then regulatory RNA. We'll talk mostly about the first three. Messenger RNA is what does the transcription of the DNA code. So what happens within that nucleus is the DNA is surrounded by what's called a Drude force field, by the way. It's kind of like this. It's water, hydrogen bonding of water molecules around the DNA. It's like a little protective bubble, kind of. If that gets penetrated, then we have DNA things going on. So that, double helix DNA strand will be opened up whenever the signal is given, okay, will be unwound, and then this messenger RNA molecule, which is like a piece or a strand of nucleotides strung together, that complements that DNA molecule, okay, whatever it's trying to read or pick up the message of. This is the transcription process. Now, from there, that's going to leave the nucleus through nuclear pores in the nuclear membrane and go into the cytoplasm where most of the rest of the stuff is done. Some of it may be done in the nucleus as well. But then that message gets through transfer RNA molecule will get translated. So we have transcription done through messenger RNA and translation through the transfer RNA. And then we have ribosomal RNA, which is really the one we're going to say is responsible for the production of the amino acid sequence that's produced outside of it. So it's like taking the language of DNA and then putting it into a language, translating it into the language of proteins. So we have DNA or nucleotide language being translated into protein language or amino acid language. I mentioned the polarity of water in a couple slides up, okay? This is highly relevant when we're talking solutions. Water is a polar molecule. Remember, in one of our first slides in this presentation, we saw the oxygen, and you can see it here again, oxygen molecule kind of on one end where the hydrogen molecules pulled to the other end. This is polarized. We have the oxygen carrying or holding a more negative charge in relation or contrast to the hydrogen end of the molecule, which is going to hold a more positive charge. Due to this difference in charges from one end of the molecule to the other, it is a polar molecule. Polar molecules will want to dance with other polar molecules, or it will make it easy for them to dissociate and bond with one another. I mentioned water having, hydrogen bonding properties. Hydrogen bonding is a van der Waals force that's not super strong, kind of a bond between molecules. But what it delivers, I mentioned that it delivers that surface tension. That's why the water strider can do what it does. But it also delivers a high heat capacity, which means that water, the substance of water, if it's pure water, can tolerate a lot of heat before it changes its state from like liquid to gas. Okay, if you're boiling water, it can hold or tolerate a lot of heat. This is relevant in physiology because so much of you, depending on where you're at in the body, 60 to 80-ish% of you is water. So your ability to hold heat and then transfer it somewhere else has a lot to do with this type of force. Also, due to the polarity, I mentioned this, of the molecule, we'll go back to that for a second, and these hydrogen bonds, water will act as a solvent. And what that means is what I've already said. It's a polar molecule. It's going to want to dance with, interact with other polar things, other charged particles. So if you have salt in water, salt ionically bonded is cation and anion. So that molecule itself is a polar molecule. When we put that in water, water will dissociate the salt and kind of rip that molecule, be able to rip that molecule apart. One thing that is highly relevant in physiology are acids and bases. I already mentioned this when I was talking about the denaturing of proteins. This is huge. One of the biggest things your body is always doing is regulating the pH of the blood primarily, but and this is done through the use of what's called a buffer. Okay, so you have an acid. Let me talk you through what an acid is. What an acid is, it means that there is a high concentration of hydrogen ions floating around in a solution. What this does, if something is highly acidic, it has the ability to dissociate, dissolve, rip things apart. If you can picture in your head a pH scale, right? The pH scale reflects acidity or alkalinity, something being basic, okay? It is a logarithmic scale of how we look at this. So if something is highly acidic, meaning it has a lot of hydrogen ions in concentration, it will be low on the pH scale. Great example is hydrochloric acid, one of the acids in your body. It's in your stomach. So that being said, that type of substance has the ability to help break down other macromolecules in food. On the opposite end of the spectrum, we have a base. A base has hydroxide ions in solution in a heavier concentration. This would be the higher end of the pH scale. So, for example, ammonia. Let's say ammonia, okay? Ammonia is highly basic, therefore It has a higher concentration of hydroxide ion in solution and is higher on the pH scale. Now, the way that we change the pH of something is utilize a buffer, and this is at play in you constantly. A buffer, particularly in your body, is called the bicarbonate buffer system, is one of the most heavily employed in your body. A buffer is something that applied either to a base or an acid is going to bring the solution, be able to bring the solution to neutral or closer to neutral. In the body, the pH range of our blood should be 7.35. That is on the pH scale, to 7.45. Anything outside of that range is going to start to change things. When we have abnormal pHs within the blood, something that delivers higher acidity will be called acidosis, and something that delivers a more basic environment will be alkalosis. Now, again, I think it's just easier to get into acidosis. One of the concerns of like a hardcore ketogenic diet or diabetic ketoacidosis is that when the body begins to break down fats, it changes the pH of the body, and it doesn't take much to throw a person into a state of ketosis. Now, it can be harmful if it goes on too long, or the body can't regulate the system quite right. Your first buffer system is your respiratory system. Your part of your breathing. Okay, we understand that there's an oxygen carbon dioxide exchange within the breathing process and the respiratory system. What that is doing beyond those two things is also buffering the blood. By doing that, it is buffering the blood. So it's bringing anything that's highly acidic back to neutral or highly alkalotic back to neutral. The kidneys are the last in line. Okay, so respiratory system, primary buffer system, kidneys, I would say, are secondary. So again, whatever doesn't get taken care of by the lungs is going to be pushed onto the kidneys to try to regulate the pH and buffer the system. Okay, in this part of the lecture, we're going to go into the parts of the cell. Hopefully, this is review for many of you. Remember that within a cell, we are eukaryotic cells, okay, meaning we have membrane-bound organelles. And our DNA is encapsulated within a nucleus. Those are the biggest differences between two of the biggest differences between us and bacteria. The cell has a membrane, the plasma membrane, which is a phospholipid bilayer. Remember those phospholipids we learned about? Well, it is two layers put together with the little tails of those phospholipids facing towards the inner side of the membrane. Now, this membrane is highly relevant in the semi-permeability of the cell, what gets in and what gets out. We just walked through briefly about the polarity of water, and this membrane has a lot to do with polarity. If due to polarity something can't move through the membrane, or maybe due to the size or some other factor, there are going to be proteins embedded within that membrane that are going to help move things through. Right on the inner side of the membrane, we begin what's called the cytoskeleton. Cytoskeleton is super cool. I think it's a protein network with different size protein strands, 3 different sizes. And it will help anchor that membrane into the rest of the cell. It serves as like a communication pathway or like zip lines. But it's like the skeleton of the cell. And it moves too. It doesn't stay stationary. Other parts of the cell will include, and you guys have probably heard of a lot of this, the endoplasmic reticulum. There's what we call a rough ER and a smooth ER, each with slightly different functions. The rough endoplasmic reticulum is going to sit outside of the nucleus. It has ribosomes on it. Ribosomes are protein, little protein manufacturing factories. Okay. The smooth endoplasmic reticulum will be a continuation of these membranes. Outside of that, outside of the smooth endoplasmic reticulum, then is the Golgi apparatus, and it's this stack of like red tube looking things on top of one another with the little vesicles hanging out on its edges. Now the Golgi apparatus, if you see that and you need to define what it is and what it does, it's the refinement center for a lot of things. One thing I want to mention is that cells have what we call an endomembranous system. It's a continuation of membranes, and along the, it's like a conveyor belt, okay, and along the conveyor belt at different points, the function changes slightly. So along with, you know, in that endoplasmic reticulum, where in the rough ER, if we know that ribosomes make proteins, the little dots, and you're going to find those on the rough ER, you're going to find them on the nucleus, you're going to find them maybe in the nucleus, you're going to find them throughout the cytoplasm of the cell. There's going to be a high level of protein production in that area, and that makes sense. And then as you go out into the smooth ER, you're going to add maybe some carbohydrate, maybe a little bit of lipid production as well, and some detoxification. Within that Golgi, again, this is kind of the refinement center. It will send off vesicles, these little red blobs, okay, send them through the cell to the appropriate place to be, whether, you know, if it's junk that needs shipped out of the cell, will be shipped out. If it's something that needs shipped to, you know, replace pieces of the membrane, it will do so. Wherever it needs to go, it's going to make its way there. Now, the mitochondria of the cell, we probably all know this is the powerhouse of the cell. This is where the Krebs cycle takes place, or your primary center for energy production. Within the cytoplasm of the cell, which is like, it's going to be a jelly-like fluid, okay? The cytoplasm includes the cytosol as well as the cytoskeleton, so the fluid and then the framework or the network. One part about the nucleus that I didn't mention, within the nucleus, okay, let's talk about the nucleus 1 second. Within the center here, we have the nucleolus. You're going to have chromatin, which is going to be part of the genetic code replication process in DNA and RNA. Okay, the nucleus is surrounded by a double phospholipid bilayer. So remember, the cell membrane, the plasma membrane, is made of one membrane that's a phospholipid bilayer. The nucleus has two, and it has holes in it called nuclear pores. I mentioned that when I was talking about the RNA molecules leaving the nucleus through the nuclear pore. That's what I was talking about. And I think that's pretty, oh, I have to mention lysosomes. Lysosomes are like the stomach. A lot of digestion takes place in a lysosome. A peroxisome, if I had a favorite organelle, it would be peroxisome, because this guy's going to take care of some detoxification things, primarily hydrogen peroxide, which is present in a lot of metabolic processes within cells, but it's toxic to the cell, so it's going to break it down into its non-toxic components. Then we have the centrosome. The centrosome is an area that does not have a boundary, really, like not a defined boundary. Within it, though, are the centrioles. These are protein filaments that lie perpendicular to one another, pretty close to the nucleus because they're going to be involved in setting up the spindle apparatus during cell replication. Here's that plasma membrane that I was talking about. So just walking through it, here's our phospholipid bilayer. Okay, here, the little balls are the hydrophilic heads. Remember, hydrophilicity means it loves water. On the inner side of the bilayer, the tails are tucked in. Okay, these are hydrophobic. They don't like water, therefore they have to be the nonpolar end. Here we have different types of proteins. Out here would be the extracellular fluid, and inside here would be the intracellular fluid or space. Here we have what's called a transmembrane protein, or an integral protein goes the whole way through the membrane, but we might have what is called a peripheral protein, which does not go the entire way through the membrane. Is going to be on one side or the other. These guys will serve as transporters of molecules in or out of the cell. Also, you might have these little green things sticking up are really important. These are connected to proteins within the membrane. These little green, this whole entire thing is called a glycoprotein. Glycoproteins will be relevant in the formation of what's called the glycocalyx. The glycocalyx deals with cell identification. That's going to be relevant when it comes to immunity, or like if you have to do a blood transfusion or something. Okay, these guys will help in the determination of foreign or self. So super important. These little blue things within the plasma membrane, these are cholesterol molecules. I mentioned that previously. They help give the plasma membrane a little bit more form, not so fluid, and a little bit of rigidity, which will be important. Now, on the inner side of this membrane in the ICF, we can see these are actin filaments, or small size protein filaments that are going to help anchor the membrane into the rest of the cytoskeleton within the cell. So that membrane, let's talk about membrane transport and different types of things that can happen. And why it's important. Okay, so first of all, we have two different kinds of transport, either passive or active. Passive transport does not require energy for anything to pass through. This would be diffusion or osmosis. Now, when we talk about diffusion, anything regarding diffusion means that we're going to have particles on both sides of a membrane, okay? And the concentration of one side is going to be different than the concentration of the other. So what happens in diffusion, it will always go with or down the gradient, meaning high concentration will move particles across the membrane to the area of lower concentration. When we talk about osmosis, osmosis is the movement of water through the membrane. Now, osmosis will happen in conjunction with, or if diffusion cannot happen. Diffusion may, struggle a little bit if the size of the particles, if particles are too big, or if there's a polarity difference, or there's a million reasons why water would move rather than the particle. When we talk about active transport, this requires ATP, or energy, in order to move things from one side of that membrane to the other side. This includes the sodium-potassium pump. Every cell in your body utilizes sodium and potassium all of the time. And about 50% of all of your energy goes into the operation of this pump. Now, what this does is it helps with the creation of what we would call an action potential, or really electricity. We're moving charged particles from one side of a membrane to the other side, and in the doing so, this is partly responsible for electrical activity. Let's talk about viruses a little bit. Viruses. Okay, first of all, there are two theories in virology. One is what we're probably most used to. However, there is another theory. Okay. There's the germ theory, meaning we get infected by things outside of us. There's also the terrain theory, which is slightly different. It's probably two-part, if I understand it correctly, that the strength of the terrain, meaning your body, immune system, high function, you're going to have a better chance at being able to combat any viruses that come at you. However, there's another theory in virology, and I do believe it's under the terrain theory or part of the terrain theory, which states this: viruses are actually blow off from our damaged cells, which honestly makes a lot of sense if you understand like what they look like and how they work, because they're just weird little alien things. Okay, we don't classify them as living because they don't have all the parts to be alive. But it's like a membrane bound ball of genetic material. So it's interesting. And there's, variations of that as well. But the way that they reproduce is by getting into the cell and then utilizing, resources to make more of what they are, right? Which then rolls downhill and creates some weird symptoms within the body. They may have a DNA center or an RNA center. Retrovirus makes DNA copies of its RNA. So I mentioned the cytoskeleton, which is super cool, which is a network of proteins, like I said, like a zip line. Super cool. It does help deliver shape and give strength, but also highly used for communication. Mitochondria, already mentioned, the powerhouse of the cell. The Krebs cycle happens here. Let me talk through some of the parts of that mitochondria. Within it, you'll notice folds, those folds, okay? Those folds within it are going to deliver more surface area so that energy can be created across those folds, across that membrane in there, okay? Those things or those folds are called cristae. Within or in between them, we have the mitochondrial matrix. Again, this is mostly where the large portion of your aerobic respiration takes place. When we talk energy production, we have to understand ATP. And ATP does fit into the nucleic acid category, okay, just due to its construction. It has a nitrogenous base, a sugar, and some phosphate groups. Now, ATP is produced through a process called phosphorylation, the addition of a phosphate group onto a molecule called ADP. And there are other molecules that are used in the body to make energy or utilized for the harvesting of energy. The way that we get energy, and I would guess, or our assumption at this point, is that this molecule is utilized and harvested because it's easy. It's easy to break apart and it's easy to rebuild. So that we're designed just to do this really quickly and easily. So I mentioned glycolysis. Glycolysis is an anaerobic process that happens in all cells, and it's the first step. And it happens within the cytoplasm of the cell. It will produce pyruvic acid molecules, which will be sent down to the mitochondria for the rest of the process, which is the citric acid cycle and that electron transport chain. The citric acid cycle has a product of a couple ATPs, okay, but also NADH and FADH2, which are what we would call coenzymes, okay, and these are derivatives of your B vitamins. So if you don't have proper B vitamin nutrients on board adequately and all of the time, these processes are not going to happen super great, okay, because the way that things are utilized, recycled, required, you need them to be able to catalyze some of their reactions. The electron transport chain is where the bulk of your ATP is produced, and it happens across those membranes, those cristae within the mitochondria. I mentioned the Golgi apparatus, that stack in the picture that we saw a few slides back. It was that stack of red-looking pancake discs between the smooth ER and the plasma membrane. It will produce vesicles, like I mentioned, of varying sorts with different things in them, and send them to where things need to go within the cell. I already talked through ribosomes, the different kinds of things in the or the rough ER compared to the smooth ER. And I talked through the nucleus. This is a great way to say this too. Within that nucleus, I mentioned the presence of chromatin. I don't want to get into that too deeply, okay? But when a cell is not dividing, we will find inside the nucleus the chromatin. When the cell is in the process of replicating, then that chromatin will be condensed, and then the chromosomes will be visible. So I already mentioned this as well. The DNA serves as the map to deliver to RNA in order to finally come into protein production. So we have the language of DNA. It is transcribed and translated by RNA into the language of proteins or amino acids. The making of RNA from DNA, okay, this is messenger RNA doing the transcription process of the DNA code within the nucleus. Again, I was mentioning that double helix will break apart. It used something called RNA polymerase first to bind itself to the DNA, make a little replication bubble within which this process can happen. That strand of messenger RNA comes up into the transcription bubble, or the replication bubble, and it will begin that formation of matching the nitrogenous bases together. Okay, so when that messenger RNA molecule gets sent out for translation, then it's going to be in the form of what's called a codon, okay? And it has to be, you have to have like 3 base pairs in a row to make a codon, which means it's going to be in a language that can be translated into an amino acid. An anticodon, you have codons that will deliver a language to produce a particular amino acid, and the anticodon is going to help with the stopping of the production of certain things. I'm not going to go into that too deep. I'm going to allow you to read that in your book. I don't think that goes into it super deep either. Now, when we go into cell division, okay, the cell cycle, this is important, I think, to look at pretty deeply for us as we understand human health. This is a big deal. So interphase is the most of what the cell will be doing most of the time. Okay, a cell will be resting and building itself, sort of nesting before it has a baby. The mitotic phase is when the cell is actually going through active replication. Now, in this picture, and it really depends on the resource you look at, cytokinesis is when the cells actually split. Some resources say that is a separate event from mitosis, others included in it. This one, it includes it in the entire mitotic phase. So within interphase, we have something called G1, or cell maintenance and growth. Then we have a part of interphase called the S phase, or the synthesis phase. This is when DNA is replicated. Then we have a G2 portion of interphase. This is, again, another phase of maintenance and growth, and when we're really checking things over to make sure we can go into mitosis. Now, during the whole cell cycle, there are multiple places where the cell has what's called a checkpoint. In the G1 phase, we will have a G1 checkpoint. That's making sure everything is on point and ready, getting the cell ready to rock and roll into the next phase. The G2 checkpoint is right before mitosis. Again, cell is checking everything. It will shut down if something is wrong. What if something went wrong in the DNA synthesis part? Okay, it needs to double check that DNA is in good shape. Otherwise, we're going to have issues. There is another checkpoint in the metaphase portion of mitosis also to make sure all the chromosomes line up appropriately. It's not noted in this picture, but that's a super relevant one as well. Now, within the mitotic phase itself, that is divided into processes. Prophase is when the nuclear envelope of the cell begins to break down, the chromatin begins to condense so that you can see it in definable chromosomes. Metaphase is when the chromosomes line up on an imaginary line in the center of the nucleus called the metaphase plate. Then anaphase is the portion of the mitotic phase where the spindle apparatus will separate sister chromatids to opposing poles of the cell or of the nucleus. In telophase, this is where the cell or the nuclear membrane begins to pinch in the center, or the cell begins to pinch in the center, And then actual cytokinesis is the full pinching and separation. Why that is all relevant, particularly those checkpoints. If at one of the checkpoints something is damaged and you have certain chromosomes that are responsible for managing this, cancer can get out of control and mitotic division will be crazy. That's one of the problems. Now, normal cells also may have a thing in place called apoptosis, where it's programmed cell death. Okay, if something's wrong, it's just going to commit suicide, sort of. Abnormal cells, they grow more, they grow where they don't belong, and they are not programmed for cell death. That's the danger. They have an awkward mechanism that we have a hard time understanding and controlling. One thing I want to mention about cytokinesis is that a key word will be cleavage furrow, okay, when the pinching really begins. And this picture here takes you through each phase of mitosis. One thing I've neglected to mention about prophase is you have the setup of the spindle apparatus. Remember those centrioles I mentioned within the centrosome of the cell during mitosis, okay, those protein filament structures that work perpendicular to one another, just sitting outside the nucleus now, are separating and going toward opposite ends of the pole. And they're going to do that so those chromosomes can line up along their network of zipline protein fibers across the cell. So cell differentiation, I'm not going to get too deep into this for our purposes, but it is important to note going through physiology that cellular differentiation is quite the cool process. It's one of the things that I think it's fascinating. They start from similar stem cell and then get told what to be when they grow up. So we're going to notice this in different areas of development in an organ or organ system. Super relevant here, gene expression. Not all genes are active in all cells. What I want to impress here is that only about 20% of your entire genetic code, okay, is responsible for or expressed, okay? Let me rephrase that. If you have any conditions that you don't like, okay, only 20% of it comes from genes, okay? Most of your genes but don't get expressed, which is interesting. To our knowledge, they don't get expressed. Now, there's something in genetics called epigenetics that says something like this. It takes cellular environment to be perfect for a gene to express itself. That is true. Oftentimes in health, we see that a particular disease, disorder, or condition will manifest and express itself maybe as a person ages or in a particular gender. That's interesting. Those things, okay, if it's a gender thing, there's a hormone difference, right? Age is likely the same thing. The hormones begin to change. So these guys can impact genetic expression to either manifest something or not manifest something. And then the question remains, can something that has been expressed, can we wrap it back up? You know what I mean? And unexpress and pick something different. That would be cool. And I think we're working on that. Free radicals and oxidative stress. This is highly relevant, I feel, in physiology. Honestly, it's not a hard concept. And then again, this relates to that pH issue, really. It is linked to that. So in terms of cellular environment, if we can understand the impact that free radicals have and then how to manage that better so that we don't have high levels of oxidative stress in our body. So a free radical is a highly reactive chemical, something with a charge, right, that wants to dance with something else in our body. So it causes damage to a cell because it wants to take something from it. We manage free radicals and oxidative stress by antioxidating agents, which are generally vitamins and minerals to suck up these guys and reduce the reaction potential. Okay, let's go through some of the medical terminology. Something that is cancer producing is a carcinogen. Something that contains 3 glyceride molecules would be a triglyceride. So if you know the answers to these, what is an ion with a negative charge? And then the second question, what is a protein with a carbohydrate group attached? I mentioned it really quickly as we were looking at the plasma membrane. This will be looked at deeper in your text. The ion with a negative charge is known as an anion, and the protein with the carbohydrate group attached is called a glycoprotein. Okay, what are the fundamental particles of an atom? They are protons, neutrons, and electrons. And what are the 3 basic types of atomic bonds? Those are covalent, ionic, and hydrogen. What happens to the human body when an unstable isotope undergoes radioactive decay? I didn't go deep into that part. That can be reflected more in your textbook, but I mentioned the radioactive isotopes for carbon dating. When that energy gets released from these type of molecules, atoms, it would be atoms, they can damage cells and cause illness, but they can be used to destroy the cancer cell. Not really selective in what they're harming, right? They'll harm health. Radioactivity will harm healthy cells as well as cancer cells. The main purpose of carbohydrates in the body: energy or fuel. What determines the shape of a protein, and what are the 4 levels of protein structure? The shape is determined by the sequence of amino acids it contains, and the 4 levels are primary, secondary, tertiary, and quaternary. What are the 4 types of lipids? They're fats and oils,